My name is Chao. I'm okay, now it's working. Today I'm going to talk about the optical biopsy using OCT and OCM. In my lab, we are working on developing new optical imaging technologies. And especially, I think people in this room are familiar with the standard clinical procedure of taking biopsy to evaluate disease. So basically, with the standard procedure, you take the tissue out, you stain them and cut into very thin slices, and then after looking under the microscope, you can tell whether it's benign or malignant. But everybody knows this is a very lengthy process. It takes several days or weeks even to get the result. And so what we wanted to do is to be able to get the images really quick using optical imaging method. We want to achieve optical biopsy so that we can get in situ real-time imaging of the tissue microstructure with a resolution that's comparable to histology. And ultimately, if we don't really have to take the tissue out or processing them, that would be the best. And if we can achieve that, it will be very helpful, especially for situations where biopsy is hazardous. For example, for ophthalmic applications or cardiovascular imaging, you really don't want to destroy the actual tissue. And here's the outline of my talk. I'll give a brief introduction of OCT technology and show you a couple pictures how it's being used in ophthalmology. And then I'll show you some recent study we're conducting in my lab. We're using this technology to look at the heart development in fruit flies. And we also use it to image cancer in pathology lab. And we're also working on technology development to further improve the speed and the resolution of this imaging technology. So just to give you a comparison, we are familiar with ultrasound. It's widely used in all the hospitals. And with ultrasound, you can get a very good imaging depth. You can see tens of centimeters in tissue. But the drawback for ultrasound is the resolution is limited. Even with the highest frequency ultrasound, you can get about a 50 micron image resolution. And we cannot use that to see individual cells. It just doesn't have the resolution. On the other hand, people are familiar with confocal microscopy. It's widely used in biological labs, and you get a very good resolution, some micron resolution, but we all know the penetration is very limited. You are lucky if you get a 200 micron, but for most of the samples, 50 micron, 100 micron, that's what you get. So the OCT, OCM technology we are working with lies somewhere in the middle. So we can get a micron scale resolution. At the same time, we can get a 1 to 2 millimeter imaging depth. And the most beautiful thing is that we don't need to stain the sample. So the principle for OCT is similar to ultrasound. For ultrasound, you send in the sound wave and detect the back reflected sound wave. And then you can measure the time delay between the signal you send in and the signal coming back. You know where the signal is coming from. But with OCT, we are using near-infrared light. And the light travels much faster than sound. So you don't really, it's very hard to just measure the time delay of the reflected photons. So instead, we have to use special interferometric method. And then we can tell like which depth the signal is coming from. And if you focus a beam on one position, you can get depth-dependent reflectivity. And if you scan the beam in one dimension, you get a 2D cross-sectional image. And if you have a 2D raster scan, you can get an entire 3D image of the tissue with micron scale resolutions. So this OCT field has been growing very fast in the last 20 years. It was initially demonstrated in 1991 by Professor Jim Fujimoto at MIT. And after that, it takes several years to pick up. And then in the last few years, every year there are like almost 2,000 papers published in this field. The majority of them are for ophthalmology applications. And so this is a standard fundus picture if you go to an eye clinic, and that's what you see. You can see all the vessels, but that's all you can get. with OCT, we can get a nice cross-sectional images of the eye. You can see the fovea, you can see different retinal layers, and this is the optical nerve head. That's all the like blood vessels and the nerves goes into our brain. That's our blind spot. And recently, with the improvement of imaging speed and the resolution, people can really get the micron scale resolution to see individual layers of the retina, and people can also use it to look at the anterior segment to image the cornea non-invasively. And actually, by imaging the cornea, you can measure the angle between iris and the cornea, and you can use as a predictor for glaucoma. Because for glaucoma patient, the intraocular pressure is high, it's pushing the lens forward, so this angle will be reduced. Okay. And so what we do in our lab is we're taking advantage of this imaging technology. We can generate high-resolution 3D images of a fly. And we're working with a group at Harvard. So basically, they can generate flies with different genetic mutations. And actually, you may not know it, but fly has a heart, and it's on the back of the fly, and it's very relatively superficial, so we can see that with this imaging technology, and we can actually see the fly heart beating in real time. You can see when the heart is fully dilated, it's only about 100 micron in diameter, and the heart is beating at 5 or 6 hertz, so it's really fast. So in control flies, you can see nice and regular heartbeat. And if we overexpress, this is a presilin gene that's found in human Alzheimer's disease. But we found if we overexpress this gene, that can cause cardiac arrhythmia, and the heart is no longer beating regularly. And so if we further did another experiment, we knock out this gene, and you can see the heart is much smaller, it's beating slower, and it also has cardiac arrhythmia. Yeah. I just show these examples to you that we can do completely non-invasive imaging on the fly, and we actually compare different genetic group at different age, and so we can see how the heart function and the heart structure changes. And another very interesting study we are doing is, because we know fruit fly goes through a metamorphic changes. It's a larva, then become a pupa, and become an adult fly. So if you look at the structure of the fly, the heart is initially a long tube, but when it become an adult fly, it become a conical shape. So we think, okay, because since our imaging is completely non-invasive, can we follow the same specimen when it's a larva and all the way when it become adult fly through all the developmental stages? And we see some very interesting phenomena. So initially, at the second instar larva, we can see the heart beating very regularly. And that's kind of expected. And then third instar larva, the heart is a little bit bigger, and it's still pumping very regularly, 5 or 6 hertz. That's good. And then when it becomes pupa, in pupa day 1, the heart is still beating, but it's dramatically slowed down. And so that's a little bit surprising. And then the second day in pupa, so there's no heartbeat. So when we did the experiment, we were surprised. Okay, did we kill the fly or something? But so we didn't give up. So okay, let's keep monitoring fly and see what happens. And the next day, in pupa day 3, you can see some heartbeat, occasional heartbeat, but it's slow compared to like a larva. Okay, then pupa day 4, and it's still it's beating, and it's a little bit faster than pupa day 3. And when the same pupa emerge and become an adult fly, we can see the heart is beating very, very fast again. So this is a very interesting phenomenon, and we repeat it in like 30 specimens, and we always see this interesting, very nice trend. The heart rate was very high, about 300 beats per minute, and significantly reduced the heart rate and drop down to 0 and stay at 0 for more than a day. And then it gradually increases, and when it becomes adult fly, the heart rate is 300, 400 beats per minute. So we don't fully understand what's going on. And we were working with our collaborator. We kind of knock out this, it's a circadian gene called the krypton. And when we knock that out, we see the heart rate was significantly different in late pupa stages. And our collaborator, they also run some genetic analysis. They look at the RNA expression level at different different pupa stages. They found out actually when the heart was completely stopped, there are certain genes that's overexpressed by 1 billion times. I think there are more than 100 genes that's overexpressed by over 1,000 times, and some genes were underexpressed significantly. So it correlated very well with our heart function information. We are doing more study trying to understand how this could happen. Okay, so another area we are working on is to use this technology for cancer imaging. And so we have done some imaging on fresh human specimens, excised human specimens. On top is an image we get with OCT and OCM. And on the bottom is a corresponding histology. This is a normal human thyroid. And as you can see, you can see this nice round follicle structure in the thyroid. And when you zoom in, you can even see individual nuclei from those thyroid. So here is a case of thyroid cancer. This is a papillary carcinoma, and you can see you don't see those normal round follicles anymore, and you see these dark regions which correspond to calcification formed in the cancer. And when you zoom in with high resolution, you can see the papillary tube formed in the cancer. And another type of cancer, this is a follicular variant papillary carcinoma. We can see the boundary between, there's a little bit normal tissue here, and this is micro follicles in the cancer, and this is a capsule between the cancer and the normal region. And we can also get the nice 3D images of the cancer. This is round, so this is a normal follicle, this is a microfollicle structure, and this is a tubular structure, papillary tubular structure formed in papillary carcinoma. Okay, we also did images in human breast tissue, and these are normal breast lobules and normal duct and adipose tissue, and these are the breast tumors, and these invasive tumor, and this is in situ tumor. Lobular carcinoma in situ. And so we can also stitch all the high-resolution images together and image a large region. This is about a centimeter by a centimeter region. It can be acquired in about 10 minutes. And all of these can be done without staining. Just fresh tissue, you can image very rapidly, you get the result. You don't have to wait for histology. You don't have to section the specimen. And I want to show you like this region, you can see the corresponding histology. We see the adipose tissue here. There's some lobular structure in the middle. This is a big cyst. And we can also look at this region. So these are some normal lobules and some adipose scattered around. And here's another group of lobules in the middle that's connected to collagen tissue. Okay, actually there are some, we can put the data into a format like Google Earth. We can zoom in, zoom out, and pan around and see different regions with high resolution, high pixel densities. Okay. And this is an example of invasive ductal carcinoma, the OCM image and the corresponding histology. You can see group of tumors over here, and these are the connective tissue between the tumors. Okay. Here are some normal kidney structure, and this is a glomeruli, and these are the tubule structure in the kidney, and these are clear cell carcinoma and the papillary renal cell carcinoma. So different. So we actually asked 3 different pathologists. We give them some training set, and then give them evaluation set, ask them to read the OCM images with blinded from histology result, and they get a pretty good sensitivity and specificity in differentiate benign images from malignant tissues. So we get very, very good accuracy. And on another hand, in our lab, we are working on keep pushing this technology forward. One aspect we are working on is to improve the imaging speed, as I mentioned before. So for traditional OCT image, if you want to get a 3D high resolution image of the eye, you are moving your optical beam back and forth, and to get a large coverage of the retina. So each 3D data set can easily take you like 10 seconds. And on the fast scanning axis, it's okay. You can still get a high resolution, good, nice cross-sectional image. But on the slow scanning axis, you're susceptible to a lot of motion artifacts. Just because it's it's very hard for the patient to keep their eyes still for 10 seconds. Even for me, that's very challenging. And our approach is that instead of doing single beam imaging, we can do multi-beam imaging, and using parallel imaging, we can significantly improve the speed. So the concept is straightforward, but when you really try to implement it, it's not that easy. You may say, okay, we can build a 16 OCT system, but it's not practical because the cost will be 16 times or even higher because you have to think about how to synchronize different systems, right? And our approach was, instead of using a single optical fiber in the sample arm, we can split the light into multiple fibers, and we can put different optical delays into each channel. And each delay will create a very short time delay on the order of nanosecond, but that's enough to separate the signal coming back from different sample locations. So we call this space division multiplexing. And with that, we can use the same light source, same detection system, basically without increasing the cost of good, without only increase a few $100 because these are very mature telecom components. And we can significantly improve the performance of the system. So just to show you an example, we can do the first demonstration we had, we can image 8 different sample locations simultaneously. In less than 0.4 seconds, we can get an entire 3D scan of the fruit fly. So that's more than 20 times faster than what can be done using current commercial system. And we can also look at a different segment of the heart simultaneously and see the dynamics of the heart beating. So you can see this segment contract first, and this segment follows that, and there's time delay between the A5 and A6. You know which direction the heart is beating from this analysis. So so the take-home message is very simple. So there are optical imaging technologies out there. It's called OCT, OCM, that we can use it to get in vivo 3D structural and functional images of the tissue with a micron scale resolution and the millimeter scale imaging penetration. We don't really need to remove the tissue or process them. Okay, I would like to thank my group members and collaborators, as well as my funding sources, and thank you very much for your attention.
Just for one question, if anybody has any.
Are you thinking about using this interoperative?
Yes, yes.
Because there you don't have the penetration problem in general.
Right, you just see the surface, yeah.
And that's particularly useful for things like breast cancer.
Mm-hmm, right.
Where you could take a look at that, look at and you don't even have to use red light.
Right. Exactly. This is actually one direction I'm very interested in, but there's a lot of engineering work that needs to go in here. Yeah. 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 Thank you. Mm-hmm. Mm-hmm. Right.
But my question to you is, when you say you have installed this that have high sensitivity, are you asking them to look at the images generated by OCT?
Mm-hmm. Yes.
And they're making a diagnosis. I assume that's what they're asking.
Yes, they are making. Right. They are. Yeah. The diagnosis is not to tell the subtype of the tumor. It's just a benign and malignant. Just to give a. Yes. Yeah. Mm-hmm. All right. Thank you.
Thank you.